When you hear about sequencing someone's DNA, it sounds like you're sequencing the entire genome, right? So front cover to back cover, but that's typically not what you're actually doing because there's a lot of information in there that's highly repetitive that makes it so that it is really computationally and financially expensive in order to do that. And you're not getting a lot of added information when you're sequencing all of this excess DNA that has all this sort of repetitive information that scientists aren't even sure does stuff. And so often, instead of doing that whole genome sequencing, that WGS, scientists will do whole exome sequencing, WES. And here they're just looking at the protein coding regions of the genome. So the parts of the genome that actually have the instructions for making proteins. Especially in the former days, people were like, oh, well, if someone has a mutation that's associated with some sort of disease, that mutation must be in that exome. It must be in the part that actually makes protein, because when you have a mutation in the protein making part, then you can have a change in the protein that causes the disease. And so scientists sometimes would do exome sequencing, but then they're like, okay, well, there's no mutation that we can find, even though it seems to be similar to this. And so then they might do the more broader genome sequencing and find mutations in things like regions for regulating the usage of that gene or the processing of that gene. But a lot of times people will do this like exome sequencing. And that'll typically be a good first start. And it's going to be a lot cheaper and easier because you're not having to have all these repetitive regions that then you're trying to say, okay, well, does this go here or here or here? And so that whole exome sequencing kind of gets around that. However, it's still going to be really expensive and time consuming. And so instead of doing that even whole exome sequencing, scientists will often do more targeted sequencing. So where they're going and looking at specific genes or specific locations in the genome. Take, for example, you do one of those at-home DNA tests, and I'm not going to go into the ethics and everything with that, but what they're actually doing is they're often doing a SNP chip. So they're not doing any sequencing at all. Instead, they're just looking at defined locations and seeing what SNPs or single nucleotide polymorphisms you have at that site. So a SNP, SNP, is a single nucleotide polymorphism. So it's a single letter change in the DNA, and so that might cause a change in a protein, it might not, and it's typically just going to be some sort of natural variation that is associated with something. And so it's not that it necessarily causes something, but it's correlated with something. So maybe it's correlated with a population from this area of the world, or maybe it's correlated with an increased risk for this thing, but often it's just because it's nearby something. But so they have these wells with the different things, that SNPs that they're testing for, and then they can just go and see which of these SNPs that you have without actually sequencing anything, just by seeing what binds. And that's going to be a lot cheaper.